Unit 4 for IAL Biology. We're preparing here for the exams in January. Let me show you a brief overview of what we're doing today. First lesson in Unit 4 talks about photosynthesis. This is something you've probably done in your IGCSE. You must have done it. It's talking about plants. How do they make their own food? So we're going to divide today's lesson into 6 parts. One is easy. One is going to be revision. So the beginning is just going to be revision to what you have done in IGCSE. Then we're going to be working on something you have done in Unit 2. Unit 2 about chloroplast and its structure. We have a couple of practicals. One is about photosynthesis, directly about the effect of light on photosynthesis. And the other one is a bit of chemistry. It's called chromatography, if you've done chemistry before. Then we have another part here where it gets a bit more challenging. We are going to go into depth into the chloroplast to see how they actually do photosynthesis. So as I stated, I'm going to start with the basics so we can all be on the same page. I'm going to show you the overall reaction of photosynthesis. So it all starts with CO2 combined with water. Together, CO2 and water, they form glucose and oxygen. This reaction can only occur in the presence of a catalyst, that's chlorophyll, and with a supply of light, which comes from the sun or any source of light, as we're going to see later on. Okay? So that's what they wanted us to see at the beginning. They wanted us just to have a clear overview of the reaction of photosynthesis. What I want to do here is I want to show you where do these raw materials, where do they come from? So let's start with the water. The water comes from the roots by osmosis, okay? So there's water in the ground, the plant absorbs that through the roots by osmosis, and then it has to climb all the way to the leaves. And that's going to be done by means of vessels. Do you guys still remember we call those vessels that carry water? Those are known as xylem vessels. So you have phloem and xylem. The xylem is going to be the one to carry the water all the way up to the leaves. Second, I need the CO2. CO2 is going to diffuse into the leaves by diffusion, all right? Stomata into the leaves so that it could combine with the carbon dioxide. Main product is glucose, and we're going to produce oxygen. Just like carbon dioxide, oxygen will diffuse out of the leaf. Okay? So something quite important, which is this, listen, check the elements on the right side of the equation. What elements do you have? You have carbon, hydrogen, oxygen, right? It's quite important to understand where does each element in glucose come from. Now, carbon dioxide provides me with the carbon. I have color-coded the elements. So carbon dioxide, the carbon in the glucose comes from carbon dioxide. The hydrogen in glucose, which is now important, comes from the water. And the oxygen in glucose, listen to this one, the oxygen in glucose comes from the carbon dioxide, not from the water. So the oxygen in the glucose, the source of it is CO2. That's not the water. This comes very often in multiple choice. You will realize later what happens to that oxygen. Do you see that oxygen here? That oxygen in the water is going to be wasted. That's the waste material of photosynthesis. So one more time. In my equation, you get you get that oxygen, and that oxygen comes from the splitting of water. So simply, we're going to take that water here, we're going to split it into hydrogen, oxygen. The hydrogen is so useful, so precious, but the oxygen is going to be a waste of, it's going to be a waste of material. Okay, great. Also, something quite important here, which is ATP. You've heard of ATP, but today I'm just going to show you the chemistry of ATP. How is it made and how is it used? So you probably know that ATP is, in biology, is like the units of energy. It's like what you do in physics. In physics, you use joules uh, for energy. Here we use ATP. And ATP comes in a recyclable manner, meaning it could be charged like this or could be uncharged. And that's something very important about ATP. That's what makes it very useful for any biological process. It could be charged up, it could be broken down. Now, how or what is the, like, if I am to look at the chemistry of ATP, how does it look like? It looks quite similar to nucleotides, unit 1. Do you remember how nucleotides look like? They're made of a sugar, pentose. They got a nitrogen base like this, right? In addition, I got 1, 2, 3 phosphates. So again, it's basically a nucleotide with 3 phosphates attached to it. Look at the way I'm going to show you that last bond in ATP. Do you see that last bond here? This is quite very, very rich with energy. If I break it, I will end up with a lot of energy. If I make it, then I'll be storing a lot of energy. So I could simply do like this, and I released energy, or I could make it and store energy for later on. I want to remind you of 2 words. One word, to build up. Do you remember that chemical reactions you took in unit 1 when you're building up larger molecule starts with C? And we have another reaction where we actually breaking down, and that starts with H, right? So ATP can be broken down or built up. So this is me taking ATP and breaking it down. That is hydrolysis. So again, ATP, you add water to it and you release energy by breaking that last bond I was telling you about. I can also build up ATP by the opposite reaction. So I told you it's a condensation reaction, but the reaction I have here is a special condensation reaction. We call that phosphorylation. To phosphorylate ADP, this is by the addition of phosphate and by making this bond, the last bond, the one is rich with energy. Okay? What did we learn so far? We've been learning about ATP. How does it look like? You can sketch it like this. It's a nitrogen base, like this, right? That's here, attached to a sugar, a pentose sugar with 1, 2, 3 phosphates. And that last bond here is what matters. You can build up, you can attach that phosphate, and that's called phosphorylation or condensation. And you can break it down, okay? Now, plants have their own way. They have their own way of making ATP. Let me show you how. So look, this is photosynthesis, right? We make glucose. That glucose we've just made has so many ways of being used. You could possibly store it. When I store glucose, I make starch. So that's number 1. The second way, I could possibly make building blocks like cellulose, right? Remember, cellulose is like beta glucose. I could possibly turn it to amino acids, but how? Amino acids got CHO and there's an N. So by adding nitrogen, nitrates from the soil to the glucose, you can make amino acids. And you could also turn it to lipids. I'm going to show you all of this next class, next week. But what I'm concerned here about is that the same glucose we've just made here will be used for respiration, right? We talk a lot about photosynthesis in plants that we forget that plants actually respire. So that glucose that plants make, some of it will be respired, used in the mitochondria to release energy, to release ATP. Okay? You have any questions? Just raise your hand if you have any question, or text me here in the chat box because I have them right here in front of me. You have any question? Tell me. Right. A bit of chemistry. So to understand today's lesson, you must have good knowledge of chemistry. And let me show you how, or what do I need to do here. So in chemistry, we have two types of chemical reaction. There's one we call oxidation, and there's one we call reduction. The simplest way to understand oxidation reduction is in terms of hydrogen, and that's the one we're concerned about. So for today, the only definition of oxidation reduction has to do with hydrogen. How? This guy on the left, the blue one, when it gains a hydrogen, I'll say it has been reduced. And the guy on the right, when it loses a hydrogen, I would say that he has been oxidized. So again, in other words, if a particle or a molecule gains a hydrogen, it's been reduced. And if it loses hydrogen, what will I say? I'll say it's been oxidized. Now, a bit more of chemistry about hydrogen itself. Hydrogen is the simplest element in the universe because a hydrogen is just an electron and a proton. So you'll hear me sometimes today, I'm saying gain an electron, gains a proton. Either way is reduction, right? Because when it's a hydrogen, it's both a proton and an electron. If you gain any of these particles, you're actually getting reduced. Also, some little thing here is like the way I'm showing you hydrogen is like oversimplified. Like the hydrogen is not going to run or jump from one molecule to the other the way it is shown here. There's actually a dedicated molecule to do this. And this one is known as a coenzyme. What's a co is like corporation, a company. So think of a coenzyme as an enzyme helper. So this is a molecule whose job is to carry the hydrogen or the proton I was telling you about. We're going to call that molecule an ADP, right? It's like a short for a very, very long word, chemical word. You don't need that word. Just call it an ADP. And that NADP is meant for what? It's meant to carry the hydrogen from one molecule to the next one. All right? So instead of the hydrogen being carried like this guy is going to come along and transport it from one molecule to another. And again, what do we call that molecule? NADP. Okay? All good so far? All right, now I am gonna do a bit of revision because I need to remind you of how leaves look like. This is what we call a leaf blade or lamina. I'm going to show you how a leaf would look like sideways, like in terms of layer. So any leaf comes with upper epidermis, lower epidermis. That lower epidermis comes with stomata. That's where gases move in and out. In addition, you got palisade. Those are the cells that are very rich with chloroplasts. And right on top of that, the whole thing, we got a waxy cuticle. Remember, that's the one that traps water inside the leaf. Adding to this, I got the spongy mesophyll. Those are the ones that have air spaces to let gases move in and out, right? So again, a leaf like this, each layer has its own function. Plus, we got that vascular bundle, which contains what? Which contains xylem to transport water and phloem to carry sucrose, the products of photosynthesis. Okay? So let's recap. If I say transport, support, what am I meant to choose from what I have here on the right? Can you write me in the chat box what am I meant to pick here? So transport support, what is the part of the leaf that is responsible for? Sorry, it's my face. Yeah, there you go. Any clue? Yeah, so veins, right? If I say large area for light absorption, what am I meant to choose? Any clue? It's broad. That's another adaptation. Allow gases to move in and out. What am I meant to choose here? Easy, right? The ones to allow gases to move in and out is stomata. Absorbs light is chlorophyll. Allow gases to reach the cells easily. Now we know that. Do you know what that means here? Being thin means short. Do you remember this? Short diffusion distance. And that is how gases diffuse quite rapidly. Good. Now, a bit of revision from Unit 2 about the chloroplasts. In Unit 2, you took the structure of chloroplasts. I just want to show you how it was used to be before it was a chloroplast. Do you remember once I told you in Unit 2 that chloroplasts were actually an independent photosynthetic bacteria that was engulfed by one of our eukaryotic cells. So you see that cell here? This cell has engulfed a bacterium whose job to generate ATP. We call in this mitochondria nowadays. And it has also engulfed a photosynthetic bacterium, means a bacterium with chlorophyll inside. And now it's turning to what? Now it's turning to chloroplast. And that explains something to me. Take a close look. Chloroplasts have a double membrane, all right? And the reason why it has a double membrane, because when it was engulfed early in the beginning of life, it had what? It had a vesicle. We still have that vesicle right at the moment. And that vesicle is what gives the chloroplast its second membrane. So again, every chloroplast comes with outer membrane, inner membrane, in addition to what? in addition to layers and layers of membranes inside, what I call those stacks of membranes, I call these the thylakoids. All right. So again, I always imagine thylakoids as being something like this. They look just like that, like stacks and stacks of membranes, all on top of one another. Each plate got chlorophyll on it. All right. So the thylakoid is an individual membrane. All right. A stack of thylakoids is what we call a granum. And those thylakoids, they're not like independent. Do you see those yellow lines here? You see that yellow line here, yellow line here? Those are known as lamella. Lamella, they're like orient the thylakoids in the right angle to absorb as much light as possible. So again, let's say the sunrise is from east. The lamellae will tilt the thylakoids towards the sun. Near sunset, they will tilt them towards west. So that's the job of the lamella. In addition, I have some jelly material inside that chloroplast, and that's what I call the stroma. Stroma is quite important because this is where you have all enzymes required for photosynthesis. So again, let's take a close look at the chloroplast. Looks like this. At this position, I see stacks of thylakoids. White is the lamella. You see them? Okay. And there is ribosome. Do you see that ball there? That's a ribosome. There's DNA. Circular DNA reminds me of the chloroplast's origin. Okay. So all of this is there with a double membrane. Don't forget, chloroplasts come with a double membrane. We have a new word, stands for what? It's called compartmentalization. What does that mean? In biology, we're very keen on what? We're very keen on isolating structures. The more specialization, the more isolation of structures, the more efficient the process is. What I want to tell you here is that in the chloroplast, you have separate compartments. Imagine how small the chloroplast is, right? Even inside the chloroplast, we're going to subdivide the chloroplast into two main compartments. One is the stroma. And in the stroma, we're going to do what we call the light independent reaction. What's a light independent reaction? It's the part of photosynthesis that doesn't rely on light. And here in the thylakoid, we're going to do the light dependent reaction. That's the part of photosynthesis that only occurs during sunlight. So again, what I'm trying to tell you here that the chloroplast itself is going to be subdivided into two main parts. One will be responsible for capturing light. That's the part that got the chlorophyll. And one will be responsible to make the glucose, right? Capture light in the thylakoid, make glucose in the stroma. Okay? Now, I want you to try this. I want you to check this out. I have 4 letters. I'm going to try this with you. Each time I show you something, you're meant to write me in the chat box either A, B, C, or D. Okay? So when I say stroma, which letter are you guys gonna write? Ribosomes are 70S. So yeah, for stroma, which is the most appropriate letter for stroma? Stroma is that jelly stuff, right? So C. where do I have the word thylakoid? Which letter is pointing at thylakoid? Good job. Which letter to make, quite tricky this one, which one to make photosynthetic enzymes, to make them, not to store them. Where in the cell do you actually make enzymes? Where do you make proteins? Make, not code for proteins. You make proteins on the ribosomes. So those ribosomes is where I make my proteins. So here. Absorbs light energy. What am I gonna be using here to absorb light energy? That is what? The thylakoid. A 70S ribosome, I've said it, right? Listen to this one, codes for the enzymes. So the chloroplast is so independent that it codes for its own enzymes. And that's going to be that circular DNA. Carbon fixation. Where do you do the fixation of carbon? I didn't tell you about this, but that is when we make the glucose. That's the light independent. Yeah, where do you do the light independent? You do it right here. Lastly, enzymes for light independent reactions. So where would you have the enzymes required for the light? I broke something. Required for the light independent reaction. That's going to be here in the stroma. Okay? All good? Okay. So I have one question here for you. I want you to prove that you're still around and tell me what's the correct answer. They wanted you to label Z. Do you see Z? Can you label Z? Choose the correct letter. You want me to zoom in? Here's Z. What do I call a stack of thylakoids? A stack of thylakoids is a granum. So that's a granum. The red is stroma. Sorry, the red is lamella, I should say. And the blue is stroma here. That's stroma. Okay. All good? Right. We're now gonna start our first practical for today. The first practical talks about photosynthetic pigments. So I'm gonna show you something. Near autumn, you start to see leaves in these beautiful colors. Now, why these colors only appear in autumn? Why do we see the orange, the yellow? The reason is those are minor accessory pigments, right? So again, an accessory pigment is a pigment that helps in photosynthesis. They're already there. They're always there. Throughout the year, they're there. But chlorophyll is in way higher amounts that we see leaves throughout the year in green, unless in autumn, when the plants realizes something. What does the plant realize near autumn? The day is getting shorter. There's no point of me wasting energy having all that chlorophyll. So instead of doing that, I would just minimize the amount of chlorophyll and get rid of my leaves. So again, those accessory pigments are known as carotenoids. because of their color. So again, you have two major pigments, chlorophyll A, chlorophyll B. Those are the ones mostly responsible to do photosynthesis. And you have accessory pigments. We call them carotenoids. Those help the plant. How? You know, like light from physics, light has so many wavelengths. And the plants are keen to absorb as much of the wavelengths of the light, the wavelengths of the spectrum. So what do these plants do? The plants basically have special pigments to absorb as much of the spectrum as possible. You can actually check this. You can actually take a plant and put colored filters. You see that purple thing? That's a filter. And we expose the plant to the light. And we see when does the plant do higher rate of photosynthesis. How do I know this? I simply check the number of bubbles. The greater the number of the bubbles, the higher the rate of photosynthesis. I'm swapping with blue. Apparently, it's doing a lot of photosynthesis. With green, check. In green, the plant is not doing much photosynthesis. What does that tell you about green? It tells you that plants don't absorb green. And if you don't absorb a color, then what we end up with, you'll end up with reflecting the color. Again, my shirt is blue. Why is it blue? It reflects blue, right? And absorbs other wavelengths of light. Same thing applies here. The reason why plants look green, because they hate the green. They reflect the green and they absorb other wavelengths of light. Okay? So by trying all these possible filters, we will end up with something like this. We call that an action spectrum. Do you see what I have on the y-axis? On the y-axis, I have rate of photosynthesis. And check, when does the plant have a high rate of photosynthesis? Near red, near blue, and very little near green. So basically, an action spectrum is the rate of photosynthesis at different wavelengths of light. So this tells you when does the plant do a lot of photosynthesis and when does it do less photosynthesis. So again, here's another graph. Here, check. This shows me what? This shows me all these pigments I was telling you about. And when do they absorb like wavelengths of light? And again, check. This tells you something quite important, that chlorophylls, as they are, they absorb a lot of wavelengths of light, but the carotenoids do help. How do they help? They allow absorption to happen at areas where chlorophylls are not good at absorption. So like, for instance, here, near 550, who's doing the best job is the carotenoids, right? Someone's asking, what's the definition of an accessory pigment? An accessory pigment are pigments other than chlorophyll who's responsible to absorb different wavelengths of light. Okay? I want to show you a question. Look at the question here. They gave me this graph. The graph shows absorption spectrum, absorption of light. You can see I got chlorophylls here, chlorophyll A, chlorophyll B. They did not show the carotenoids here. What do they want me to do? They wanted me down to draw an action spectrum. How am I supposed to do this? So again, I need to draw a graph, sketch actually, to show an action spectrum. What's the difference? That is an absorption spectrum, okay? An action spectrum is like the one I showed you here, where it shows rate of photosynthesis. What do you notice when you look at both graphs? How are these related? They're identical, right? So the absorption spectrum at the bottom and the action spectrum at the top, they are more or less the same thing. Because simply, the plants do a lot of photosynthesis when they absorb light. So my job here is to do what? Is to repeat the same graph, same pattern, not like this, like that maybe. It's a sketch. I just need to show the same trend somewhere here. So just like this, follow that, trace that same graph you have at the top and show them that it would have more or less the same pattern. Okay? Good. You follow the pattern of both. You follow the pattern of chlorophyll A and chlorophyll B. So just show both of them. You see what I've done? Okay. Next experiment is chromatography. You've taken chemistry before. Chromatography is the way we separate pigments. So in our case here, we're meant to separate pigments that you have in a leaf. But before I start this, I need to do something. I need to extract chlorophyll from a leaf. To do so, I have spinach. I'm gonna take the spinach. I'm gonna add either ethanol or acetone, because both of which dissolve chlorophyll. And then I'm gonna put sand. Why sand? Sand will give me some rough texture so I can break the cell walls of these leaves. Okay? Then I get something like a paste. That paste got all these pigments I was telling you about. What am I going to do about this paste? I'm just going to take it and put it on the baseline of a chromatography paper. Pencil baseline, right? Next, I dip this into my solvent. And again, the solvent of choice is either ethanol, acetone, okay? And then you would dip the paper like this, and you separate the colors, right? So you would see what? You would see chlorophylls, you would see some hint of orange. These are my carotenoids. So again, what did we do so far? We crushed, we filtered, we got that filtrate. Then we took a chromatography paper. We drew a baseline like this with my pencil here. And then we took that chlorophyll filtrate there. We put it on the baseline, this. And then we started to see colors like that all over. How am I supposed to identify these pigments? How do I know which pigment is which? Which one is chlorophyll A? Which one is chlorophyll B? And which one is a carotenoid? We have a special method. We call that the RF values of these colors. So you grab a ruler and you measure what? You're going to measure the tip of the spot. So for instance, here's my spot. How much is the RF of that tip? It's 14.5. I divide this by what? I divide this by what I call the solvent front, which is this here. Let's show it in black, maybe. So you can, black, I said. Black, right, black. Doesn't want to. Okay, so let's assume that this is the solvent front. What's the solvent front? That's the distance traveled by water or any solvent. So I divide the 14.5 over, let's say, the 16, and that would be your RF. And that RF tells you the identity of the pigment. So for instance, here, how much is the RF of that pigment? It's 4. It's actually 3.8. What's the solvent front? It's 4.9. So the RF is going to be something like 3.8 divided by 4.9. It's 0.78. Okay? And from this, you can identify, you just look it up on a data booklet or on the internet, and you can determine what photosynthetic pigment is this one. Okay? All right, so let's just do quick look on the specification. What did we do so far? We looked at the overall reaction. We looked at the structure of chloroplast. We checked the absorption spectrum. We did the chromatography. So what are we left with? We're left with the last 2 parts of today's lesson. One talks about phosphorylation, and one talks on hydrolysis. So we will learn now what? We will learn how do we generate ATP by photosynthesis, and we will learn how do we actually start to generate or make glucose. Okay? So just like what I told you earlier, that there are 2 types of chemical reactions take place inside the chloroplast. One we're going to call light dependent, and what are we going to call light independent. All right? So let's start with the light dependent reaction. As the name suggests, it's a reaction that would only occur if there is light. So that reaction would only occur if the plant is exposed to sunlight. During this reaction, look what happens. Water molecule gets exposed to energy. Again, the source of energy in this case is the light itself. And that water goes into a reaction where we're going to split the hydrogen from the oxygen. This reaction is known as photo. What does photo mean? Photo is light. Photolysis, in one word. Basically, you're splitting, lysing water into 2 particles. One is a waste, oxygen, and the other one is the one that really matters. Do you remember what I told you at the beginning of the class? I told you the oxygen from water is going to be wasted. So this oxygen here will soon diffuse out of the plant. We don't need it. But I need those 2 hydrogens. So we're going to split off those hydrogens from here, and we're going to make use of them. So take a look. This hydrogen is going to be taken out. This hydrogen is going to be taken out. And the oxygen will turn into O2, which is wasted. Okay? Let me show you that photolysis reaction once again. With the help of light energy, what are we going to do about the water? We're going to take the water and we're going to split it into 2 particles. One is just a waste, oxygen. Okay? And the second is hydrogen ions, H+. And the third is an electron. Do you remember what I told you at the beginning of the class? I told you a hydrogen is those 2. So every hydrogen atom is a proton and electron. Okay? So that is photolysis. What am I going to do now about these hydrogens? Do you remember this guy? The NADP, the coenzyme. He is going to take those hydrogen ions from the thylakoid, where light-dependent reaction takes place, is going to take them from here into here. So again, the first reaction took place on the thylakoid membrane. The second reaction, the light independent, is going to be in the stroma. Okay? So we're going to grab those hydrogens, collect them from here, transfer them to the stroma, and there we're going to take the carbon dioxide like this, and I'm going to add those hydrogens to the carbon dioxide. Guess what's going to be the outcome? Hydrogen, carbon, oxygen. Does that remind you of any molecule we started the class with? Hydrogen, carbon, oxygen. This will soon become what? This will soon become a glucose, right? So let's recap. Photosynthesis is two reactions. One I'm going to call light-dependent reaction, and one I'm going to call light-independent reaction. The light-dependent takes place in the thylakoid, while the independent takes place in the stroma. During the light-dependent, we take a water and we split it into 1, 2, 3. H plus, 2 electrons, because I have 2 hydrogens, and 1 oxygen atom, or half an oxygen molecule. I'm balancing the equation here, okay? Then, with the help of the carrier, we transfer those H plus from the thylakoid into the stroma. We combine them with carbon dioxide, and we generate the glucose. This is like a bit oversimplified, but that's the essence, or that's like what photosynthesis is all about. I'm only missing one thing here. You know what it is? For this reaction here, I need an input of energy. The input of energy was light here, right? That's why it's called light dependent. But the light, you can't simply take light energy and carry it from one place to another, right? Doesn't make sense. For that reason, I will convert that light energy into ATP here, and I will use that ATP for this reaction. So to sum up, the light-dependent reaction gives me two things. Gives me those precious hydrogens. It gives me ATP, which are required for the light-independent reactions. Okay, I'll repeat. This is what photosynthesis is all about. We divided photosynthesis into 2 stages. One is called light-dependent reaction, and one is called light-independent reaction. The light-dependent is on the thylakoid, the light-independent is in the stroma, and it's a cycle, meaning the products of the light-dependent are used in the light-independent. The products of light-independent are going to be used there with the light-dependent. Let me show you how. So we said during the light-dependent, we split the water. Do you guys still remember what we call it when you split a water? We called it photolysis. Photolysis. We split the water into what? Oxygen, a waste, right? In addition, I'm gonna take the hydrogen ions with the help of this guy. Do you remember his name? NADP. I'm actually gonna call it reduced NADP. Why do I call it reduced NADP? Because it gained the hydrogen. It's coming from here to here, and an ATP. Okay? Those would be taken to the stroma. In the stroma, we're gonna take a carbon dioxide. So I need my carbon dioxide. I lost my carbon dioxide. It's here, right? And I'm gonna reduce that CO2 into glucose. Once I'm done with the light dependent, I'm gonna take back what? ADP and NADP without the hydrogen. Okay? If you could sketch this along with me, it would be very helpful for you to remember. So again, a chloroplast we divide it into two parts. The one on the left is going to be the thylakoid. Okay? And the one on the right is going to be stroma. Okay? Here, my input is water and the output is oxygen. Okay? On the right side, my input is carbon dioxide and the output is glucose. And in between, we have materials moving left and right. This way, you have NADP with the hydrogen. And the way back is going to be what? It's just going to be an NADP minus the hydrogen. So we recycle it back. Okay? And then we also require another important molecule, which is ATP. So you have ATP going this way, and you have ADP going the other way around. Okay? Someone's asking, does the NADP carry the hydrogen? Yes. Its job is to carry the hydrogen ions. Okay? Let's try something. Let's try a question. They gave me chloroplast, and they said, complete the diagram to show structures involved in light dependent. So what am I meant to draw here? They said light dependent. Do I draw the stroma? No. I meant to draw what? I'm going to draw thylakoids. Like this. Another set of thylakoids. I can link them up, this, with lamella. Okay? So that's a granum. And the red one I have here is one thylakoid. Okay? Someone's asking, what's the job of ATP? Look, my friend, this reaction is quite very energy consuming. It needs an input of energy. ATP will provide me with the energy required to reduce, listen to the word, to reduce carbon dioxide, because that carbon dioxide will gain hydrogen. All right? Let's do quick comparison between light dependent and light independent, okay? Can you help me? Could you, in the chat box, put D here and I for this, okay? Someone's asking, what does ADP stand for? My friend, do you remember when we were talking about ATP right here? We said at that time that ATP could look something like this with 3 phosphates, right? Or with 2 phosphates, right? Adenosine diphosphate, 2 phosphates, or adenosine triphosphate. So the ADP version of it is the one that doesn't have enough energy, okay? The charged version, the one that has a lot of energy, is ATP. And the one that doesn't have enough energy, what am I going to call that one? We call it ADP, right? So let's make that comparison, dependent versus independent. Again, in the chat box, tell me D or I based on what I'm saying. Again, when water is split, when did we split? When did we do photolysis? Remember? We did it on the left. We did it in the light dependent, right? Where did we make ATP? When did we make that ADP? T, with a T, right? We made it on the left. We consumed it on the right. Where did we hydrolyze the ATP? Where did we break it down? Where did we make use of it? It's right here. Which reaction takes place in the stroma? which one of those two reactions happens inside the stroma? It was independent, right? Where did we convert? What does fixation mean? Fixation is when you convert gaseous carbon dioxide into solid glucose. So that is here, right? Thylakoid, which reaction happens in the thylakoid? It was this. Where did we make the glucose? We made it in the dependent or independent? Here. Where did we attach the hydrogen to the NADP? Where did we make the reduced NADP? Here, right? Where did we remove the hydrogen? Remember, if you take off or remove a hydrogen, then it is oxidation. Okay, all good? Right. Now, today we're dedicating the remaining part of today's lesson for the light dependent. So my job now is to focus on light dependent. Next time, next week, we're going to be working on light independent. Okay? So let's just focus on what happens on the thylakoid. So look, I'm going to take the thylakoid just like this, one of them, and I'm going to split it into two parts. So this part is what I'm going to call thylakoid membrane. And the inner part, which is shown right here in yellow, I'm going to call thylakoid space. Okay? So the thylakoid itself can be divided into two parts, the membrane itself and what's enclosed inside that membrane. Okay? So here we go. So here's the first event. First event is when the light hits the chlorophyll. You see that molecule here? That's a molecule of chlorophyll. We usually describe this as a photosystem. Photosystem. Why we call it a photosystem? Because it's not only chlorophyll, what I have here, it's chlorophyll with the accessory pigment. So that bunch of different pigments all together is what I call a photosystem. Okay? So what's going to happen now? As you hit that chlorophyll with the light energy, you will excite some of the electrons you have in the chlorophyll. What does it mean when I'm saying like an electron's been excited? Usually, if you give energy to a molecule, you would notice that some of those electrons in the molecule, they gain energy and they jump out of that molecule. You see this clearly when you hit a piece of metal, like aluminium, with light. What do you notice? You notice it shines. The shining of that metal or the shining of the molecule is when the electrons jump out of that molecule. So an electron jumps like this, and it goes from being at a high energy stage. Look, it's just like an object. I'm lifting up that object to a higher stage, and then it drops down slowly, like this, right? Every time you have an object, you lift up and you drop down, what will it happen? Here, at this position, it has potential energy because it's been lifted up. As it drops, it gives me back energy. You know, like in physics, we always say that energy is never wasted or never made. Same thing here. That light energy I gained, it was used for what? It was used to excite the electron and use that energy here to move the electron down. But while this electron is moving down, it's not wasting any energy. How? Because while it's moving down, I'll be using that energy to pump hydrogen ions from where to where? From the stroma all the way inside the thylakoid space. So see, I'm packing hydrogen ions inside the thylakoid space. For what reason? Soon I will let those hydrogen ions out like this through a very narrow channel, like the one I have here. And I'll be using that energy that I've stored there for what? You see what I've done? This is me packing all these hydrogen ions inside the space here, inside thylakoid space. And then I'm going to let them run through a small channel here. As they move, we're going to use that energy to generate what? We're going to use that energy to generate ATP. All right. One more time. What have we done here? It's like this. We did several things. We took energy from the sun. We excited some electron, like the one I have here. And we moved that electron along like a chain. While this electron is moving, it gives me energy. That energy is good enough for what? It's good enough to pack and pack hydrogen ions inside the thylakoid space. Just like this. Look. Right at this moment, they have so much potential energy inside the space. And just like this, if you ever seen like an electric station like the one I have here, the water has a lot of potential energy. If I could just let it drop down, it would be used to do what? It would be used to generate electricity. I don't want to generate electricity here. That channel that I'm using here, it will be used to generate ATP. So while the hydrogens are flowing down from their higher potential to lower potential, that energy will be used to generate ATP. Okay? This process, this thing, when I'm building up that concentration of hydrogen ions and I'm using that to generate ATP, is what we call chemiosmosis. Why chemiosmosis? Chemiosmosis because it's both a chemical gradient. I have a lot of hydrogen ion, that's chemical, right? And osmosis because I have also an charge gradient. So again, let me show you the definition. That's the definition of chemiosmosis. To move H plus to other side of the membrane and build a gradient. That gradient are good enough for what? That gradient is good enough to generate ATP. Okay? I think it's a bit complicated, so that's why I want to show you this one more time. And hopefully this time you will fully understand this. So again, try to understand where am I? I am basically here in one of those thylakoids, okay? What am I going to have to do? First, I'll expose this photosystem. I'm going to expose it to light. So I'm just going to bring some light energy and expose that photosystem to light. Any molecule in life comes with electrons. So that electron here is built in the chlorophyll. What's going to happen to this electron? It's going to run along a chain. Guess what I'm going to call that chain? I'm going to call that chain an electron transport chain. Okay? The electron is moving like this, but as it moves, it's not wasting any energy. That energy, while the electron is moving down here, is going to be used for what? It's going to be used to build a gradient of hydrogen ions, just like this. Soon, this thylakoid space will be packed with hydrogen ions. So they will run out through this channel. As they run out, what's going to happen? That will help me to generate ATP. So this channel has a name. The name suggests what it does. It's called ATP synthase. ATP synthase means it makes ATP. All right? So now I'm making ATP here. And remember that ATP will soon be used for what? To fix carbon dioxide, to make glucose. But you may tell me, like, what's going to happen to all these hydrogen ions? Like, where are they going? Yeah, so like we got them out from here. And where are they going to go? This guy is going to come along. Do you remember the carrier? It's going to take those hydrogen ions and run with them. Where is it going to run? Together, they will run out to the stroma. All right? So let's repeat. Event number one, what was the first event? Event number one is when you excite the chlorophyll or photosystem, when you excite it with light energy. You will get an electron out from the chlorophyll, right? It goes through that electron transport chain until where? Until here. Then, once we reach here, we're going to pump hydrogen ions inside that thylakoid space. Those hydrogen ions will start moving out through the channel. Remember the name of the channel? We called it ATP synthase. And there we will be using that energy to make ATP. Okay? The last thing we need to mention here is like that photosystem here will soon run out of electrons, right? We don't have like unlimited amount of electrons. Who's there to replenish those electrons? Here comes the water. Remember the photolysis thing? Photolysis will always help me to supply the photosystem with electrons. Okay? So let me show you this. Take a look, please. Here is where I start. So event number one is the photosystem here on the left. It's receiving light, okay? As it receives light, the light energy will excite an electron, okay? The electron will soon leave the photosystem and run through the electron transport chain. Meanwhile, to replace the electron, a water molecule is splitting, okay? Those electrons will now run in the chain, the electron transport chain. You see the black things just came here? That's the electron transport chain. And then as this is happening, I'm pumping hydrogen ions into the thylakoid space. All right? Now it's packed with hydrogen ions. I'm going to now soon let those hydrogen ions out. You see that purple tree there on the right? That's ATP synthase. This will now be used for what? This will now be used to generate ATP. As they're flowing out, that will be helping me to make ATP. One last time, that's what I have here. It's called non-cyclic phosphorylation. Let me show you what it means. First event, light hits that molecule. I'm going to call that molecule a photosystem 2. And this one here, I'm going to call photosystem 1. Both are the same thing. Both are chlorophylls. This receives the light, excites an electron. This is what I call an ETC, electron transport chain. This runs the electron like this. And meanwhile, those hydrogen ions are packing up inside like this. Soon, the hydrogen ions will leak out from here maybe. And as they leak out, that will help me to generate ATP. Okay? What's the job of NADP? The job of NADP is to snatch all these hydrogen ions and take them to the stroma. Okay? Let's try something. Let's try to organize our thoughts together and put those events in their correct order. I want you to take a look at this list and try to find what is the first event. Can you send me one letter to represent the first event in this cycle? Think about it. Where did we start? It all started with light energy, right? Hitting a chlorophyll molecule, just like this, okay? What happened next? Who's there to replace that missing electron? So again, what happened next that helped me to replace it? So water split. So here I have electron lost, right? And then the water replaces the electrons been lost, right? Next, think about it. What's going to happen next? Now I have the electron running in the electron transport chain. The electron move along the ETC, right? And then once it moved there, it would be used for moving hydrogen ions, right? Next, we will grab those hydrogen ions, just like this, grab them, this, and we're going to use them to make ATP. Okay, let's try this now, like visually. Okay, here's the same thing. That's what I call phosphorylation, non-cyclic phosphorylation. Look where I'm going to start. It's quite confusing that the first chlorophyll we start with is known as photosystem 2. Why do I start with 2 and not with 1? I start with photosystem 2 because they discovered this photosystem 1 first, and then they discovered this. But when you're writing the events, you start with 2. You start with photosystem 2. That's where it all starts. Okay? So photosystem 2 gets light energy, and that light energy is going to be used for what? It's going to be used to run electrons here. Where is this going to go to? It's going to go all the way to an electron transport chain. Meanwhile, that energy is going to be used for what? It's going to be used to pass these electrons to another photosystem. We excite the electron again, and we go to another electron transport chain. Next, who's going to come and collect all of my electrons? It's going to be this. The NADP takes these electrons and the hydrogens and form NADPH. All right? This the ultimate aim of this reaction is for what? Is to phosphorylate. So I take ADP, I combine it with a phosphate here, and I make an entire molecule of ATP, right? So this is the entire event. Someone's asking, how does the water replace the lost electrons? Remember, splitting of water here gives me what? Gives me 2 electrons, 2 protons, and an oxygen, right? Those electrons will provide the chlorophyll. Since the chlorophyll lost the electron, it's going to provide it with these missing electrons. Okay? Now, this is the major, this what we call non-cyclic phosphorylation is the major type of photosynthesis. Majority of plants do this, okay? Some plants, in some occasions, or some bacteria, do what we call cyclic. So again, this is called non-cyclic. Why is it non-cyclic? Because the electrons soon will be taken away, right? With the NADP. But I have another version. We call it cyclic. It's rare to happen, but it does happen. And the ultimate aim of this is just to make ATP. So this has nothing to do with making of NADPH. This will never make you any glucose. Just if the plant is short of ATP, it would do this particular cycle. Look, very simple. Look, it starts with an electron running along a chain, electron transport chain. The electron will soon return back to the beginning. It's like a circuit. Goes like this and returns back to its own photosystem, which is photosystem 1 only here, right? So as you can see here, the reason why it's called cyclic, because the electron is not taken away. So I don't have NADPH here. I don't have any photolysis at all. I don't have photosystem 2. It's just like a small cycle whose aim to do what? The aim here is to pack H plus inside and generate ATP. Only that. Okay? So again, what is cyclic phosphorylation? It is a process where electrons return back to PS1. We don't make any NADPH. We don't make any oxygen. No photolysis, nothing except for making ATP. Okay? So let me show you that cyclic again. It starts with photosystem 1. I put photosystem here especially to show you that is not involved. It involves what? An electron. It runs like this. Where to? It goes to the electron transport chain. And then the same electron will be returned back to its beginning. Meanwhile, I am using energy to combine phosphate with ADP to make what? To make ATP. Do you guys see that little I here next to the P? What does that stand for? Like this one here. Like, why do we say PI? Why do we just say P? That PI here, it's just like to tell you that this phosphate is inorganic. Inorganic stands like it's a phosphate coming from the soil, right? It's not like a phosphate you would find in any molecule in the living thing. It's just like a formality. So again, what's that picture in front of you? That's called cyclic phosphorylation. Okay? Let's compare between them. Let's compare cyclic with non-cyclic here. So again, if you believe it belongs to cyclic, put C. If you believe it belongs to non-cyclic, put an N. Can we start? So, which one involves photolysis? Can you tell which one of those events involves photolysis? It was what? Photolysis only occurs in non-cyclic, okay? Which one is aiming to make ATP? Which one just to make ATP, nothing else? Right? Cyclic, right? Which one? ATP synthase is involved. This one's tricky, right? Where did I use ATP synthase? I've used it in both. And here I make ATP, and here I make ATP, right? So both, you got the same enzyme. Where do I have a gradient? Where do I make a gradient of H plus ions? I do so in both too, right? Where do I involve two photosystems? Where do we actually have two photosystems? We have it in non-cyclic, right? We have photosystem 1 and 2. Where is the NADP? Is the final acceptor? Where do we have NADP been involved? It's actually here. And where is light energy is used to excite electrons? Both are light-dependent reactions, right? Since they're both dependent reactions, then it's in both too, right? So let's check this diagram. That's non-cyclic, okay? Can we label those molecules just to sum up what we've done? So, could you guys tell me what's the name of this reaction? What do we call reaction K? That's when I am splitting a water. What did we call this reaction again? Reactions known as photolysis. What's the waste product? Here I need to write the waste product of photolysis. You know what is it called? It's oxygen. And then the water here is passing on other product. This product is called P and is going to the photosystem. Can you name that product that jumps from water to the photosystem? What do we supply the photosystem with? We supply the photosystem with an electron. And what do we supply the NADPH with here? You know, the splitting of water gives me electron and also protons. So here is the proton. Okay? Next, here they wanted me to name that reaction. The reaction where I make ATP. Do you know what I'm calling this reaction? Since we took it at the beginning of the class. We said it's named as phosphorylation. So it's known as phosphorylation, right? But since it's involving light, we're going to call it what? We're going to call it photophosphorylation. So again, phosphorylation, you make an ATP normally, but here we use a light energy to make ATP, so we're going to call that photophosphorylation. All right? One last diagram, and then we stop. So here's again, light. They wanted me to name D. So what am I going to name D? The thing that absorbs light. I'm going to call that a photosystem. That's cyclic, by the way. Okay? So with cyclic, light hits the photosystem 1. It takes the electrons up, up, up. And then we run along this chain. Do you remember what I call that chain that carries electrons? We call it electron transport chain. I'm going to call it ETC, okay? And then during this reaction, we do photophosphorylation. To do so, what do I require? I require ADP with phosphate, inorganic. Together, they make ATP. Okay, that's cyclic. Then they said, name one molecule that is produced in non-cyclic that is not produced by cyclic. Again, in this process, there is one missing molecule that is not being produced. This molecule, remember we made a table here? There's one molecule made by cyclic, but it's not made by non-cyclic. What's that missing molecule? The molecule here is known as NADPH, or you may call it oxygen, because you don't make any oxygen here. Okay? So this means we're fully done with today's lesson. Could you send me a message on WhatsApp right after the class, so I can send you a link to Google Classroom, okay? You can watch the recording and you can check the homework, okay? For now, I'm going to let you go.